Welcome to Experimental Physiology Meeting Reports, where Dr. Christopher Lockery and Dr. Gillian Gray discuss their symposium on in vivo assessment of cardiac function in rodents.
My name is Dr. Christopher Lockery, and I'm a cardiac physiologist from the University of Glasgow.
And I'm Dr. Gillian Gray from the Centre for Cardiovascular Science in Edinburgh.
Together, Gillian and I had a fantastic opportunity to organise a symposium at the Physiological Society Annual Conference on the 5th of July 2012 in Edinburgh. The aim of the symposium was to highlight the technological advances over the last 5 to 10 years, which have enabled us to better than ever before utilise rodent models of cardiovascular disease to study the structure and function of the heart. It was extremely important to us in organising this symposium that we invited speakers who both had first-hand experience of in vivo cardiac phenotyping, but also had been at the forefront of the technological advancements which have been made to enable the field to overcome the difficulties of the small size and fast heart rate of rodents. Passing on this first-hand knowledge from users is undoubtedly fundamental to the successful establishment of these techniques in other laboratories. And as anyone who has tried to set up a new in vivo technique in their lab knows, communication of the pitfalls and limitations of a technique can save months, if not years, of valuable time. It is clear from the symposium proceedings, which are now published in the Journal of Experimental Physiology, who kindly sponsored the event, that a step increase in technology has been made over the last 5 to 10 years. This advance has substantially overcome many of the difficulties associated with assessing in vivo cardiovascular function in rodent disease models. The wide range of techniques presented in the symposium enable the cardiovascular research community to characterize cardiac structure and function with unprecedented accuracy. And it is clear that if we combine these new technologies with an integrative approach in relevant models of cardiac disease, we can put into context the impact of specific molecular changes on whole heart function and ultimately advance our knowledge on the pathophysiology of cardiovascular disease.
The symposium was kicked off by Stefan Lennart with his elegant demonstration of how the latest in super-resolution imaging techniques can be applied from the subcellular to the whole animal level, providing novel insights into sarcoplasmic reticulum-mediated calcium release in health and in disease. We then moved on to ultrasound and magnetic resonance, which are already well established as tools in preclinical imaging. However, in this symposium we heard from ultrasound physicist Carmel Moran on how new developments in high-resolution ultrasound are now allowing more rapid and detailed assessment of cardiac structure and function in utero as well as in adult mice. Craig Lygate shared with us the expertise gained by the Oxford Experimental MRI Group in assessing myocardial metabolic pathways, leading to the identification of potential new therapeutic targets during the development of heart failure. Additional insights into cardiac function can be gained from interventional studies, and James Clark from King's College focused on the pros and cons of using intraventricular catheterization to achieve this in murine models. Molecular imaging in vivo is rapidly progressing, and Adelaide Greco was able to show us how you can now use positron emission tomography to provide an effective means of assessing infarct injury in mice, particularly when it's combined with anatomical information from computed tomography. I then completed the session by showing how we're using fluorescence molecular tomography in the Edinburgh Preclinical Imaging Unit to interrogate inflammatory processes in vivo during infarct healing, adding to the structural and functional information gained from magnetic resonance imaging and high-resolution ultrasound in the same animals.